we're live.
Okay, thank you all for coming to the inaugural Early Career Immunology Seminar. I'm Tim O'Sullivan, an assistant professor at Immunology at UCLA. Before we start today, I want to take a moment to acknowledge the tragic shootings of yesterday. We understand not all of us may be in an ideal headspace. We appreciate you being here, given the circumstances. Although these continue to be challenging times, the enthusiastic support of the digital immunology community gave this seminar its initial momentum, and I want to thank each of you viewing today and the organizing committee for your efforts on social media to amplify our message and make this seminar series a reality. The purpose of this seminar series is to increase the exposure of early career faculty in a time in which we haven't achieved equity of representation at scientific conferences and seminars despite equally innovative discoveries. So even in spite of all these ongoing obstacles, we can form a movement to be the change that we want to see in our community. And we can do this by showing support for junior immunologists at critical stages in their career and giving other underrepresented immunologists a platform to share their work. To our established colleagues in the audience today, we hope that you join our mission to promote the voices of diverse junior faculty across the United States. If you sit on an organizing committee, please discuss how you can intentionally invite more junior female and underrepresented minority scientists to speak about their work. All it takes is one person to start the discussion. So now I'll step off my soapbox and introduce our first speaker, Graham McVicker. Graham received his Bachelor of Science degree in computer science from the University of British Columbia and subsequently worked on the Ensemble gene browser at the European Bioinformatics Institute. He did his PhD with Phil Green at the University of Washington, where he researched how selection shapes human genetic variation. He then subsequently did his postdoc with Jonathan Pritchard at the University of Chicago and Stanford. And here, Graham studied how genetic variation affects gene regulation in chromatin. Since 2016, Graham's been an assistant professor at the Salk Institute for Biological Studies. Graham's lab uses a combination of naturally occurring genetic variation and high-throughput genome perturbations to discover new regulatory sequences and interpret non-coding genetic variation. Graham has developed a lot of sophisticated computational and statistical methods to extract subtle signals from experimental data, which I currently nerd out about. So today, Graham is going to be talking about how a tiling deletion screen discovered some novel regulatory sequences for GATA3. If you have a question for Graham at any point, please type it into the live chat on your right, and he will address as many questions as possible at the end of the talk. So I'll read him your questions, and then Graham will answer as many of these for the time that we have allotted. This talk is recorded and will be available on our YouTube channel shortly after we finish today. So with that, I'll let Graham take it away.
Okay, thanks, Tim, for a great introduction. And I'm very happy to be able to be the first speaker of this early career immunology seminar series. It's a great honor to be the first speaker. And like Tim said, today I'm going to talk about my lab's recent work on trying to discover regulatory elements for the gene GATA3. So the focus of my lab is to interpret trait-associated non-coding human genetic variation. And to do this, we use really two main approaches. One approach is to rely upon natural genetic variation. This is genetic variation that's just out there in the human population. And we do this simply by looking for associations between genotypes and things like gene expression in specific cell types, or associations between genotype and chromatin accessibility, for example. Now, the other approach that we're trying to use to understand non-coding genetic variation is to use directed genome perturbations. So we can now use Crispr to direct perturbations to specific locations in the genome, and then try to understand how those perturbations are affecting various cellular phenotypes, such as gene expression or cellular proliferation. So I've divided today's talk into three parts. In the first part, I'm going to give some background on allergic disease genetics and how that relates to GATA3. In the second part, I'm going to talk about an analysis method that we developed for tiling Crispr screens, which we call relics. And then the third part, I'm going to put the first two parts together and talk about how we performed a tiling deletion screen for GATA3. And the first part and the third part are work that was led by a postdoc in my laboratory, Shou Chen. And the middle part is work that was led by a PhD student who just recently graduated in my laboratory named Patrick Thiao. So I'll start on the first part. I'll provide some background on allergic disease genetics and tell you how that connects to GATA3 gene regulation. So allergic disease is actually a collection of different diseases. These diseases include asthma, eczema, and allergic rhinitis. This is just what many people have: allergic rhinitis who have seasonal allergies or allergic allergies to things like cats or pollen. And all these diseases are thought to be closely connected. And part of the reason is that, for example, when epidemiologists have looked at people with asthma, they find that about 90% of people who suffer from asthma also suffer from one of either eczema or allergic rhinitis. And the close connections between these diseases is supported by genetic studies. So what I'm showing you here is a kind of meta-analysis of large-scale genome-wide association studies, looking at associations between genetics and allergic diseases. This is published by Ferreira et al. In 2017. And what I've highlighted here are just the genetic correlations. So these are the estimated amount of variance of the genetics which is shared between these diseases. And you can see that, for example, asthma has pretty strong genetic correlations with allergic diseases as a whole, as well as eczema. And so this really shows that a large part of the genetics of these different allergic diseases is overlapping, although it's not completely overlapping. They also have independent genetic components. So these are results from a large-scale allergic disease genome-wide association study by Zhu et al. In 2018. What I'm showing you here is a Manhattan plot across the genome. So here are chromosome positions on the x-axis, and on the y-axis, we're showing the minus log 10 p-value of the genetic association with the trait, here the trait being allergic disease. And you can see there's a number of very significant, genome-wide significant associations scattered across the genome. And several of them are indicated, highlighting some of the known immune-related genes which are close to these genome-wide significant hits. So if we simply take these genome-wide significant hits and we look at the genes which are nearby to them, and we perform a simple Go enrichment test for the genes which are nearby to these significant hits, I'm showing you the top ranked categories that come out of this Go analysis, ranked by fold enrichment. And as you can see, all of the top categories that come out in this table, of which I'm just showing the top 4 rows, are related to differentiation of helper T cells. So CD4 T cell differentiation. This is pretty striking. And I think it clearly indicates the sort of central role of helper T cells in allergic disease biology. So I want to go back to this Manhattan plot. And now we're going to zoom in into this one locus here on chromosome 10. It's actually 2 loci. So there's IL-2RA, which has its own GWAS hit nearby, and GATA3, which is a little bit further along the chromosome, which has actually a couple of nearby significant GWAS hits. And I want to focus specifically on GATA3 today, because it's already established to have a pretty important role in allergic diseases. And the reason it's thought to be important in allergic diseases is because it's fundamentally important for type 2 helper T cells. Now, these are the subset of activated helper T cells, which are important for defense against extracellular pathogens, such as helminths. But it's also believed that these activation of these type of T cells is also what drives allergic disease. So they're very central players in immune responses to allergens. And what's important with GATA3 in this context is that GATA3 is known to be a transcription factor, which is a master regulator of Th2 cell differentiation, and its high expression of GATA3 is required to maintain Th2 cells. Okay, so let's go back to this Manhattan plot again and zoom in on the GATA3 locus. So here I'm just showing you a 2 megabase region zoomed in around GATA3. And you can see, if you look at the GWAS signal, there's actually 2 significant risk regions which are independent from each other. So we have this first risk region, which is downstream of GATA3. And we have a second risk region, which is further away from GATA3, almost a megabase away from GATA3. And I'd also like to note that actually downstream of GATA3 is a large gene desert. So even though these hits are fairly far away from GATA3, GATA3 is by far the closest protein coding gene. There's a 3 megabase gene desert downstream of GATA3. So we can do what's called statistical fine mapping of these genome-wide association hits. And to do this, we use a tool which was developed in Matthew Stevens' lab called SUSY. And what this tool does is it tries to estimate the number of causal variants there are at each of these hit loci, as well as which variants are most likely to be causal. And so there's a lot of ambiguity about which variants are causal when you have a GWAS hit locus. This ambiguity is caused by correlations between the genotypes of genetic variants, which is caused by linkage disequilibrium. So SUSY tries to parse that out and say how many causal variants are there and which ones are most likely to be causal. So at this first risk locus, SUSY predicts that there's just likely to be a single causal variant, but there's substantial ambiguity about which variant is causal. So if we take 90%, what's called a 90% credible set, there's about 20 variants in this region, which are potentially the causal variant. Now, if we take the second risk region, it also thinks that there's only one causal variant, but it narrows it down to 3 potentially causal variants. So these are the 3 variants here, which have by far the highest, what's called posterior probability of inclusion. So we think that the causal variant here for allergic disease is very likely to be one of these 3 variants. And for most of the talk today, I'll refer back to the second risk region here of interest. So all of these observations together lead to what I think is an obvious hypothesis, and that's that these allergic disease risk variants near GATA3 affect the regulation of GATA3, and they also affect the differentiation of naive CD4 T cells into Th2 cells. So one way we can immediately try to test this hypothesis and examine it is by looking at gene expression quantitative trait locus data. So this isn't data that we generated, but we downloaded it from several databases. So we have a database of gene expression quantitative trait locus data from Blueprint, as well as from DICE, and they come from a number of different cell types. So in these original studies, what the authors did is they collected genotypes as well as gene expression data from a number of immune relevant cell types. And they just looked for genetic correlations between the genotypes of genetic variants and the expression of different genes. And in this heat map, we're just showing that representing the p-values for genetic association with the expression of GATA3 in these different cell types. And you can see here we have the 2 most significant SNPs from risk region 1 and the 3 most significant SNPs from risk region 2. And what we can see is that if we look at risk region 1, there's some evidence for an association with naive CD4 T cells and expression. And at the second risk locus, there's more of a evidence for a significant association with some of these differentiated helper T cells like Th2, Th1, and Th17. But actually, I would say, the evidence here is a little bit weak. So these p-values are just barely significant or not even quite significant in some cases. And they're limited in terms of the number of cell types which are actually looked at. And with the sample sizes in these studies, which was about 400 individuals for Blueprint and about 90 individuals for DICE, we see that the estimated effects of these gene expression quantitative trait loci, the effect of these variants on GATA3 expression is actually very weak in these cell types and not actually always significant at the current sample sizes. So we think it's desirable to have an alternative approach to try to understand the regulation of GATA3 in a systematic way, to try to interpret how these genetic variants are acting to control GATA3 expression. And so the approach that we used is to perform a deletion screen for regulatory elements for GATA3. And this is something I'm going to come back to at the end of the talk. But first, before I get to revisiting the regulation of GATA3, I want to talk about our work on analyzing tiling CRISPR screens and the method that we developed called RELIX, because this work is going to be very useful when I come back to this point of understanding GATA3 gene regulation. So this part of the presentation is work that was led by Patrick Fiao, who's a recently graduated PhD student from my laboratory. Okay, so the idea behind these genetic perturbation screens is to simply test if specific regions have, for example, an effect on gene regulation. So we might be interested in asking, does region X affect the expression of gene Y? And what we can do with CRISPR is we can simply design a guide RNA to target this region. We can direct Cas9 or some variant of Cas9 to this region with the guide RNA. And then we can ask if targeting this region affects the expression of the gene that we're interested in. And there's actually several flavors of CRISPR technologies that we can use. So the basic one is Cas9, which introduces double-strand breaks. These are repaired by non-homologous end joining, which is an error-prone process. And this results in random mutations centered that occur within around 10 to 20 bases of the target site. We can use CRISPR interference, or CRISPR-I for short. And this is a dCas9. So this is one enzymatically inactive Cas9 that doesn't introduce cuts into the DNA, but it's fused to something like a Crab domain, which induces heterochromatic silencing and transcriptional inhibition in a region spanning approximately 1 kb around the target site. We can use sort of the reverse of this, which is CRISPR activation, or CRISPR-A. And here we have a dCas9 protein fused to a domain, an activation domain like VP64, which recruits histone acetylation machinery, which results in activation of the target region. And another flavor, which I'm going to talk about later today, is to actually use a pair of guides. So you deliver 2 guides to every cell, and then Cas9 introduces nearby double-strand breaks, and you get deletion of the intervening sequence between double-strand breaks, resulting in a programmed deletion. So I talked about how you can introduce these perturbations to a single genome site, but we're interested in doing this in a very high throughput way. And there's been a number of studies in recent years which have performed these type of screens, which I'm going to refer to as tiling CRISPR screens or CRISPR regulatory screens in the context of gene expression. And so the way this works is you design guides to target across the region that you're interested in. So you have dense guides tiling across this region. You synthesize the oligos, encoding these guides. You introduce the oligos into a vector, which you can then introduce into cells. Once cells receive something like Cas9 and the guides, you can let them recover for a while, and then you can do something like subject them to selection, for example, to survival or growth. Or in our case, we're often interested in gene expression, so we can sort them into a high expression pool and a low expression pool. Once we've done this, we can take cells from each of these pools, and we can sequence the guides which are present in each of these pools. And so at the end of this, what we're left with is a table of counts that looks like this. So we have the identity of each of the guides, which were in our screen, and we have counts for the number of times we observe the guides, for example, in the low expression pool and in the high expression pool. And then the objective then is to look for sequences which affected the proportion of guides which ended up in each pool. So for example, if you knocked out a sequence which is important for the expression of a gene, you might expect guides targeting that sequence to have higher counts in the low expression pool. So our goal in developing an analysis method for this was to identify regulatory elements. And here what I'm showing you are results from a CRISPR-A screen. It was published by Simyonov et al. In 2017. And I'm showing you what are essentially the raw data from this screen, which is represented as log 2-fold change calculated in a sliding window. So each one of these points represents a different window, and we've computed log 2-fold change by simply taking the ratio of the guide counts in these windows. And I've highlighted in red the location the CD69 promoter, which you can kind of think of as a positive control in this case, because in this study, they were sorting into pools based on the expression of CD69. And so when you look at this, you can see maybe there's a number of different peaks, which might indicate that there's regulatory elements here, but actually the data are pretty hard to analyze and pick out from this. So I would say when you just look at this data, it's pretty hard to determine exactly how many regulatory elements you think that there are and where exactly they're located. So we wanted to develop a new analysis method which would help to answer these questions, both for this example screen and for any other screens that people perform. So the idea is that in RELX, I'm going to describe briefly how it works, is we segment the genome into small sequences. In our case, we typically use 100 base pair sequences, and we label the known positive controls, which I'm going to call functional sequence 0. So here's the functional sequence 0 indicated here. This is our positive control. Using this positive control sequence and all of the segments which are not part of the positive control, we can estimate what I call a sorting probability distribution. So we simply estimate how likely is it for a guide to be sorted into each of these 3 pools, for example, based on whether it targets the known functional sequence, FS0, or whether it targets a background sequence, so all of the other sequences. And so we can use the known functional sequence and all the other ones to estimate these distributions. Once we've estimated these distributions, we can then try to estimate the best location where we can place a functional sequence. So we call this next functional sequence 1. And for example, in this example for CD69, we might place it here. So we think, okay, there's likely to be another regulatory sequence there. We can then go back and re-estimate our sorting probability distributions. So update them based on the new location of this functional sequence. And we can repeat this, iteratively placing functional sequences and updating our probability distributions. And we simply repeat this process until we reach convergence and no more updates are possible. So we wanted to evaluate the performance of this algorithm using, first of all, simulations. And the reason why we want to use simulations is because there's actually not really a good ground truth data set out there for this kind of data. There is no data set where we have a systematic understanding of exactly where all the regulatory sequences are in the screening data set. So we developed a simulation framework where we know the ground truth, where we know where all the regulatory sequences are. And the simulation framework allows us to vary a number of different parameters and ask how those change the performance of our algorithm and other related algorithms. So here I'm comparing the performance of RELX and 2 other algorithms which were also designed to analyze CRISPR screens. So CRISPR-Surf, which was specifically designed for tiling CRISPR screens like these, and MAGIC, which was originally designed for the analysis of CRISPR screens that targeted protein coding genes. And so here I'm showing you the performance measured by average precision of each of the methods. How accurate are they at identifying regulatory elements in the simulation? And we're comparing the results from a large number of different simulations. So each one of these points here represents an independent simulation that we performed. And here we have 2 different parameters, which I'm calling the selection strength. So this is sort of how hard is the selection or how hard is the sorting of cells into different pools. And this turns out to be a key parameter in your screen. And here in the simulation, we're assuming that actually guides have fairly low efficiency. So often guides will have minimal or no effect. And what you can see is that under these conditions, RELX has far better performance than the competing methods. We can also simulate under medium guide efficiency and under high guide efficiency. So in all cases, especially under medium and low guide efficiency, which we think are realistic parameters, RELX has superior performance to the other methods. So at least in simulations, this demonstrates that our method works quite well. So we next applied the RELX analysis method to some published screens. So here, again, I'm revisiting the CD69 CRISPR activation screen. And these are what the actual results look like from running RELX. So here's the placement of the positive control sequence. And this is where RELX estimates all of the other sequences are located. So here, this is a posterior probability of the sequence being located at this position. We can also collapse these tracks down into a single track, which we represent this way, where each different functional sequence is represented by a different color here. And here I'm comparing the results from our predictions to the results from the competing methods, CRISPR, and MAGIC. And I think you'll agree that our results look like they show sort of a higher resolution and much more precise results than the other methods. We can also zoom in on this region of the closer to CD69. And so this is a zoomed in version of that. And so you can see here there's several functional sequences predicted by relics. And we can test each of these. So we can take the GATA cells where this original screen was performed, and we can simply sort based on the expression of CD69. And so this is a control, for example, using a non-targeting guide. This is a positive control where we've targeted with CRISPR-A the promoter of CD69. And so the expression of CD69 is increased compared to the negative control, as you would expect. We can target this predicted functional sequence 1, or it's actually sort of targeting in between FS1 and FS2. And we see, again, an increase in CD69 expression. We can target this functional sequence here that we predicted, and we see, again, this increases expression of CD69 as we predicted. And we can also target this region, which we're using as a negative prediction. So relics did not predict that there was a functional sequence here. And again, this behaves more like the negative control. So I think this demonstrates that our relics approach is doing a good job at identifying real regulatory sequences from the analysis of these CRISPR screens. Okay, so now that we've established that this relics analysis method for CRISPR screens works pretty well, we want to go ahead and apply it to our own data set, which is an analysis of GATA3. So to analyze GATA3, we chose to use what I call a tiling deletion screen. In a tiling deletion screen, you're trying to introduce deletions, and you can do this by using a pair of guides to target nearby sequences. And the idea is that you cut out the intervening sequence between where the two guides target. And you can do this for a single site, but you can also sort of tile these deletions across a region. So each cell is going to get a different pair of guides, and we can densely tile program deletions across a region. And there's several advantages to this. So I think that deletions are interesting because they're really a true knockout of the sequence versus CRISPR-I or CRISPR-A, which sort of do an epigenetic perturbation of a sequence. And they may not discover sequences which are sort of insensitive to epigenetic perturbation. Another advantage is that they can interrogate much larger regions than single guide screens, where you're just introducing small indels typically around the target sites. And the tiles also allow sort of for repeated deletions targeting a given sequence. And so that, in theory, should increase your statistical power to detect functional sequences. So we designed a tiling deletion screen to screen for regulatory elements for GATA3. We centered our screen on GATA3 and tiled individual deletions across the 2 megabase regions centered on GATA3. On average, the program deletions were 800 base pairs in length, and we designed about 15,000 guide pairs to densely tile across this region. So we designed the pairs of guides to tile across the region. We synthesized oligos containing these paired guides. We cloned the guides into a lenti CRISPR v2 backbone, made lentivirus from this library, and transduced cells with this library at a low multiplicity of infection. So each cell on average, only about 1 in 10 cells on average received the virus. And then we performed puromycin selection to only retain the cells which took up the virus. And the reason we do this is because our objective here is to have only 1 deletion per cell, and we don't want to have, in most cases, multiple deletions per cell. So after allowing cells to recover following this, we can then flow sort using an antibody against GATA3. Then we can sequence guide pairs which were integrated into the genomes of the sorted cells. And then we can analyze the resulting guide counts with relics. So in our screen, we actually perform 4 different replicates. And here I'm showing flow sorting where we do a low expression pool, a medium expression, and high expression pool. We actually did something a bit more complicated than that. So in the first 2 replicates, we decided to sort into a high, medium, and low pool as diagrammed here. We also have what we call the bulk or the input pool, which are the from the unsorted cells. But for the 3rd and 4th replicates, we added additional granularity to the sorting. So now we have a high, high, a high, a high, low, and so on. In total, we have 8 pools. And one nice feature of relics is that it's capable of jointly analyzing all of this data together. So this is sort of a QC metric for the screen. And this is some output that's provided by the relics analysis method. And what this is simply showing is a ratio of the estimated sorting probabilities for the different pools for functional sequences. So these are regulatory sequences versus the background sequences. So not sequences that don't affect regulation of GATA3. And so you can see, I think the most important column here is the low expression pool. And what this is saying is that guides which targeted, for example, GATA3 are known functional sequences, which were much more likely to end up in the low expression pool than in the background, or than background sequences were. And so you can see that guides targeting functional sequences for GATA3 were enriched in the low and the medium low pools and depleted in the high expression pools, as you might expect if the screen was working correctly. Okay, so these are what the results actually look like for our tiling deletion screen and applying relics to these data. So what I'm plotting here is sort of a raw view of the relic scores. And then here, I'm showing you the predicted functional sequences that are output by relics. So in total, relics predicts 36 functional sequences indicated by these purple bars here. And what you'll note, and GATA3 is located right in the center of this large 2 megabase region here. And what you'll immediately notice is that the vast majority of the predicted functional sequences are downstream of GATA3. And this is this gene desert region downstream of GATA3. These green genes here are actually non-coding RNAs or pseudo, and these purple ones are pseudogenes. But what you can notice here is that there's a much higher density of predicted functional sequences and what appears to be the topological associated domain containing GATA3. So this is high C data performed in the same cell line in GERCAT T cells. And you can see that this region here corresponds to what people would call the TAD, or topological associated domain, for GATA3. But there's also evidence for GATA3 making contacts, I think, for a large region which extends out for a long ways from around where GATA3 is located. And so this is kind of interesting to us. And it's also notable, I think, that we see a number of functional sequences predicted quite far downstream of GATA3. Another thing that I want to point out, which is a bit puzzling but interesting to us, is that if we look at H3K27 acetylation, which is a histone modification that's associated with active regulatory elements such as enhancers and promoters, we can see that there's a lot of H3K27 acetylation peaks in this topological associated domain, but then outside of this domain, there's actually very few. And so we have a number of functional sequence predictions here where these sequences are predicted to affect GATA3 expression when they're deleted, yet they don't look like they're sort of classic enhancers at all. So we're very curious about these. And so here I'm showing again a different view of this locus where here are the GWAS. This is again the GWAS signal for this region. And so you can see the 2 regions there. And then here I'm showing again the predicted functional sequences from relics and the relic scores, H3K27 acetylation and ataxi. And I want to just highlight a few of these functional sequences here. So we have this one, which I'm going to call FS7. This actually coincides to an enhancer which was discovered in mouse for GATA3. So this is a known enhancer in mouse T cells for GATA3, and it looks like a classic enhancer in that it has very strong H3K27 acetylation for the orthologous human sequence. We also see two other functional sequences which are located very far downstream of GATA3. I'm going to call those FS8 and FS22. And these ones do not at all overlap with either ATAC-seq or H3K27 acetylation in GERCAT cells. So we can perform validation experiments. So this is an example of a positive control where we're comparing to, this is actually a targeting negative control where we're introducing a deletion at what we call a safe harbor sequence that we don't think has any function. And you can see, as expected, deletion of one of the exons of GATA3 results in a dramatic decrease in expression of GATA3, as you would expect. Here, we see what happens when we knock out this known enhancer. So we see a decrease in expression. And now I'm going to show experiments for knockout of these two, what I would call unmarked functional sequences. So again, when we knock out FS8, we see a decrease in expression. And again, when we knock out this very distal unmarked sequence, we see a reduction in expression again, although it's more subtle than we see, for example, when we actually delete the gene itself. So we think this is very interesting. In particular, it's interesting because this FS22 sequence actually contained two of the SNPs which are predicted to be causal for GATA3 regulation. So we think this is potentially very exciting because here we have an example of what is an unmarked regulatory sequence in GERCAT cells, but it contains some of the SNPs which are most likely to be causal for allergic disease. So we think there's something potentially very interesting here where we have a novel sequence important for the regulation of GATA3. It's implicated for allergic diseases. Deletion of it does seem to reduce the expression of GATA3, yet it lacks the canonical regulatory element marks. So I will say that we performed our screen so far in GERCAT cells, which is a cell line model for T cells that's obviously imperfect. And we wanted to look wider at primary cells. So here what I'm showing are data downloaded from a study published by Soskic et al. In 2019. And I'm highlighting this known enhancer sequence, FS7, and this novel sequence that we discovered, FS22. And here I'm indicating the different cell types, which are isolated in some cases from two different donors. And looking at 16 hours post-activation or 5 days post-activation. What you can see when there's a non-specific activation, or what they're calling TH0 cells, is that maybe there's some evidence out here for a little bit of H3K27 acetylation, but not much. No evidence for H3K27 acetylation in TH1 cells or TH17 cells. But when you look in TH2 cells, actually all of a sudden, there are things that look like a very strong enhancer here, at least in two of the individuals, and maybe again, even at the early time point in this individual as well, although there's some inter-individual variation. But we think this is really interesting because essentially what we're seeing is that there's a regulatory element that doesn't look like a regulatory element in GERCAT cells, yet when we delete it in GERCAT cells, it still seems to affect the expression of GATA3. Subsequently, when cells are activated and differentiated into the TH2 subset type, all of a sudden it does look like a very strong enhancer. So that's basically where we're at with this study. This is mostly unpublished work. The RELX part has been published, but this analysis of our tiling deletion screen has not been published. We want to further characterize what's going on with these unmarked regulatory elements. It almost seems like deletion of them does affect regulation of the gene, even preceding actual marking of this region in a sort of activated cell type. So we want to perform some high chip experiments to understand the physical topology of the region. So we want to understand if this region is actually contacting the promoter, prior to being marked by H3K27 acetylation. And we also want to perform high chip before and after deletions. And we want to start moving more of our experiments into primary T cells and out of the GERCAT model. I think the GERCAT model has been great for setting up our screen, but obviously for understanding connections to human traits, it's very important to start doing more work in primary cells. So that's essentially all I have to tell you about today. So just to summarize, allergic disease GWAS clearly implicate helper T cell differentiation as probably one of the most important processes. And it specifically implicates some non-coding regions near GATA3 of interest, which we explored in some greater detail. I think the tiling deletion screens are interesting. They find a bunch of sequences that when you delete them, they do appear to affect the expression of the gene, even though they don't overlap canonical enhancer marks, at least at the time the deletion is performed. But they may overlap enhancer peaks that are sort of visible in subsequent cell types. I also described our relics analysis method. Our paper on relics has been published. It was published last fall. And you can get the relics software for free from GitHub at this URL. And if you're using it, I would encourage you, and you run into any difficulties, I would encourage you to ask us questions. We're always interested in improving it and making it better. It has numerous features, which I think are great. It can jointly analyze a large number of pools together. So in our example, we used as many as 8 pools from a single replicate. We showed with simulated data and real data that it appears to outperform all other methods that have been developed for this purpose. And it identifies both previously detected elements as well as novel functional sequences. So I'd just like to conclude by acknowledging the members of my lab. And I especially want to acknowledge Patrick Thiau, who developed the relics analysis method. And I also want to especially acknowledge Shoi Chen, who really drove the GATA3 project. So she's been a real leader on that project. And I also want to acknowledge the people who assisted us in the flow cytometry core at the Salk Institute and the next-gen sequencing core, as well as our collaborators in Bing Ren's lab, who really helped us get going with these CRISPR screens, and as well as people in my lab who contributed, who have since moved on to other locations. And this is the most recent screenshot of our lab. So this is the COVID version of our lab meeting here. And then this was the first seminar of this early career immunology seminar series, and I hope that you enjoyed it. And I also want to highlight that there's a number of other seminars that are coming up. They're going to be occurring once per month on these dates. With that, I'll conclude, and I'm happy to take any questions which were posted on YouTube.
Thank you so much, Graham. That was a really impressive talk. I mean, putting in that amount of CRISPR guides into a region is pretty impressive. So we have one question from Joe Sun, and he says he might have missed this, but does relics predict tiling CRISPR guides that have lower off-target effects compared to other prediction algorithms, or is that not a parameter of the relics program?
So that's not part of relics. So there are programs that try to estimate the specificity of CRISPR guides, as well as the efficiency of CRISPR guides. So specificity means how likely is a guide to have an off-target effect. And there's been a number of published methods for looking at that, the earliest one being by Patrick Shu. And so when we designed the guides in our screen, we used Patrick Shu's scoring algorithm to score the guides and to filter out guides which had low predicted specificity. But relics currently does not include explicit modeling of off-target effects. So it is suggested that you try to filter your guides when you design them for low off-target effects.
I kind of have a follow-up question to that as well, because we do a lot of CRISPR editing in primary immune cells in our lab currently. And one of the things that we have issues with, especially from a lot of published whole genome CRISPR screens, is that a lot of the guides don't work. And so I'm kind of curious because you've done a lot of these, what's sort of the success rate from those prediction software tools, and how do you find to maximize success with that?
Yeah, I think actually it's a huge challenge for us as well. So I think part of what makes these screens so noisy is the variability in what we call guide efficiency. And there are tools out there, a number of them now, that try to predict the guide efficiency. We think they're pretty inaccurate still. So, even though you can get a prediction, there's a lot of noise in those predictions, right? And so you can try to design your guides to have the highest efficiency as predicted by these various tools, but I still think we still find that a large number of them don't work very well. And we don't model that well. So we're making recent improvements to relics to try to explicitly model the guide efficiency, actually taking scores from these different tools and incorporate them into the relics predictions. In the current version of relics, we don't do that. And I think it's sort of, we do try to model what we call over-dispersion, which is sort of extreme variability in how well in the different guides. And I think that is accommodating for some of this variation in the guide efficiency. But if we could explicitly account for it in the model, we think it should do a little bit better still.
Okay, we have another question from Amanda asking to comment on the challenges of the transition to primary T cells and options for genes that are not easy to stain for by flow.
Right. So I think GATA3 is okay because it has a nice antibody, and so we have to do intranuclear staining to do the flow. So it does kill the cells. In our case, we don't care because we just care about sequencing the guides at the end, and we can still do that for dead cells. Genes like CD69 that was done in the Marson lab are easier because that's a cell surface marker that you can easily sort on. GATA3 is not a cell surface marker, and we were still able to do this experiment for it. But I think for a gene where you have no antibody, it becomes more challenging, although the RNA flow fish is a potential option that we haven't tried in my laboratory, but that some other laboratories have had success for. So that's where you do fish type labeling using RNA probes. And apparently that can work for flow cytometry. I forgot what the other part of the question was.
Just the challenges of transition to primary cells.
Yeah, so I think another huge challenge with primary cells is that it seems like delivery of lentivirus with sort of large payloads can be very challenging in primary T cells. So it seems like the efficiency of CRISPR-based editing or mutagenesis or any of these approaches is low when you have a very large plasmid that you want to get into lentivirus. And it seems more efficient when you have much smaller plasmids. And in our case, it's with the dual guides and Cas9, our vectors over 10 kb, I think. And so we found that doesn't work very well in primary T cells. What does seem to work a lot better is delivery of things like RNPs, so where you incubate Cas9 with guide RNAs and electroporate those into cells. And we can get efficient results doing that, but that's challenging for screening because you don't get the guides integrated into the genome then, so you can't sequence them and get the output of that. But there is work by, for example, the Marson lab, where they've done a combination approach where they introduce Cas9 by electroporation and then introduce the guides separately with lentivirus, and that apparently can work well.
To follow that up as well, like, have you tried electroporation with those fusion proteins with the dead Cas9 to look at like either activation or inhibitory just based on that in primary cells?
I don't think we've done that specific experiment yet in primary cells, but those are things that we're interested in trying out.
So are we, so I'll probably email you about that.
Yeah, I'd be totally happy to coordinate with you. Yeah, we're trying out a bunch of new electroporation techniques in my laboratory right now, so very happy to coordinate efforts in that regard.
Awesome. So we have a question from Tanya Friedman, who is asking, what kind of controls would you want to do to ensure that the stress of modifying primary cells doesn't cause a loss of identity or the function? And she's saying mostly in the context of like myeloid cells, which might be able to easily change their polarization or gene expression programs.
Yeah, I mean, I think it's a big problem. I do think, so you'll see a lot of screens or CRISPR experiments where I think people use like a non-targeting control. I don't think that's as good a control. So in our hands, at least, when there's we see a difference in cells when we have a targeting control versus a non-targeting control. And so I would encourage people to try targeting controls where you target a sequence with Cas9, but you think it doesn't have a function that's relevant to what you're looking at. And even better is probably to use a whole bunch of negative controls rather than rely on just one. But yeah, I do think negative controls are a big issue in this field right now. And knowing what a good negative control is. So I think what we've been thinking recently is that it's good to have several, and it's good to have them be targeting.
Great. Do we have any more questions in chat? Okay, well, if we don't have any more questions for Graham, this will be recorded for anyone that wanted to watch later on. I want to thank everyone for their attention today and for their questions. It was a wonderful discussion of Graham's work. Really excellent seminar, Graham. And if you have any other questions for Graham as well, feel free to ask them on the recorded YouTube channel, and Graham should be able to address those afterwards once we put that up. So
Yeah,
thank you all.
Thank you very much.
Okay. We're off.